All right, good morning, everybody. I'm sure you can hear me. Good morning. How was your night? So, without wasting time, let's start the class. I apologize for the last class that we're not able to really enjoy, maybe due to the network that was kicking me out. So, this morning we are going to move very fast. So, I want to plead with you to give your full attention. So, last week we started biomolecules and the central dogma of life. We said that these biomolecules are biological molecules that help the cell, that do certain things in the organism, and we said they are obtained from food and they are found freely in nature, and they are obtained from the food we eat. We said they are also known as polymers because they are made up of monomeric units or monomers, small subunits that come together to form a larger molecule. So please give me some minutes, I'll be back. Sorry. Okay, we said that biomolecules are made up of atoms that are covalently linked. That is, these biomolecules, they are made up of atoms that are joined by covalent bonds. We said that there are 4 major classes, of which 3 are polymers. We said of the polymers are those ones that have monomers. For protein, the monomers are amino acids that are linked by peptide bond. For carbohydrates, the subunits or the monomers are monosaccharides that are linked by glycosidic bond. For nucleic acid, the monomers are nucleotides that are connected by phosphodiester bond. And for lipids, we said lipids are not polymers, but they have 2 major subunits, the fatty acids and glycerols, and they are linked by ester bond. I don't think I mentioned that the last time. Fatty acids and glycerol make up lipids, and the bond between them is known as ester bond. Ester as in ESTER. Then what again did we say? We said the fatty acids, lipids that are saturated, and unsaturated fatty acids. And we said the saturated ones have single bond between them, while the unsaturated have a double bonds, double or triple bond between them. And we said there is a kink, a form of bend, where those double or triple bonds are formed in unsaturated fatty acids. Okay, we also talk about making and breaking of macromolecules. We said when macromolecules, when monomers come together to form polymers, so there is loss of water in a form of a dehydration or condensation reaction. And we said to form monomers back from polymers. We said this by hydrolysis. Okay, I think we should go to today's class because we have a long way to go. Let me navigate to where we stopped last time. Okay, we stopped here the last time. We said proteins have four major components: the carboxylic acid group, like your COH from organic chemistry, the amino group, the NH2 group, the H hydrogen is attached to the alpha carbon and the R side chain. And we said that this R side chain is what differentiates one amino acid from the other. All the 20 amino acids have this formula, right? They have this NH2H and COOH common to all of them. So what distinguishes them is whatever that R group is it CH3? It is that R, whatever that R is. For instance, let's say maybe R is a letter, right? Maybe the R, is it A, is it B, is it C, is it F, is it G, whatever, right? It is what determines the type of amino acid that we have. But these three other components are common to all amino acid: the NH2, the hydrogen, and the carbon, known as four major components, are attached. As we are going, if you look at this side, it has 20 major amino acids, and these amino acids are classified based on the different groups that they have, right? For instance, we have the non-polar aliphatic hard group, and you can see the distinguishing structure there is highlighted in a, do we call this color pink? Sorry, I don't know color. Maybe this is pink, right? You can see the simplest one is a glycine that has hydrogen attached to the alpha carbon, right? Then we also have beside that, we have alanine that has CH3 attached to it. If you go to the aromatic group from your organic chemistry, you know what aromatic compounds are, and you can see that ring that is attached to the aromatic R groups, right? You have the phenylalanine, the tyrosine, and the tryptophan. Then under it, we have the positively charged groups. See, like I said earlier, and histidine. All of these are structures. Like I said, they all have common NH2 amino group, the carboxylic acid group COOH, and the hydrogen that is attached to that central carbon. Then all what distinguishes one amino acid from the other is the structure that attaches itself to the alpha carbon in addition to these three major common ones. Okay, we have protein structure. Protein have different structures, right? Depending on the level at which you are structure. We have the secondary structure. We have the tertiary structure of right? Sir, please, can you slow down a bit? Okay. Sorry if I'm moving too fast, but we need to travel fast. So we have a long way to go. I don't want to engage. Okay. If I can take that again, we started from here. We said protein, major structure that comprises the carboxylic acid group, the amino group, the hydrogen group, and the R group. And we said what distinguishes one amino acid from the other is the attached side chain known as the R group. All other ones, all other components, the amino group, the hydrogen, and the COOH group are common to all the amino acids. And we said last time that the type of bond that forms between one amino acid and other to form a polymerized peptide bond, right? That is why we call amino acids as polypeptides, right? Okay, so we now got here, we said that there are 20 standard amino acids, and you can see the amino acid from the other. And please, I don't expect you to cram these 20 amino acids and these 20 amino acids and their structure, but please know the groups. For instance, if I ask you which of the following is a negatively charged amino acid, and I put the options there, know which one. They are just 20, so you can just classify them. The non-polar, the non-polar aliphatic are 6. Your glycine, alanine, valine, leucine, methionine, and isoleucine. Then for the aromatic R group, they are just 3. For the polar uncharged R group, they are 6. And for the negatively charged R group, 2. So that is that on that from that side. Then here we're talking about the protein structure, right? The protein has specific structures, right? Don't forget that in one of our previous classes, I mentioned that protein is the language that the cell understands, right? So the protein, for it to be able to do what it's supposed to do in the cell, there are conformational changes, right? Like structural changes in terms of shape that it has to undergo when it is formed, right? We are still going to go to how but protein doesn't function if it doesn't take a particular shape, right? Or those structures and what differentiates or what distinguishes one from the other. For instance, the primary structure is a linear sequence of amino acids linked together by peptide bonds, like as you can see under the primary structure here, this primary structure here, you see there are amino acids. You have your lysine, another glycine, leucine, valine, alanine, and histidine. These are amino acid residues that are linked by peptide bond. So in this form, we refer to that structure as a primary structure when they are just linked. The amino acids are just linked. We are going to see how they form, how amino acids come together one after the other to pair up. Number 1, number 2, number 3, depending on the type of protein that you have and the number of amino acids, right? Don't forget there are proteins that do different things in the body. Okay, that is a primary structure, the linear structure of the amino acids, like as they are just formed from the ribosomes. They are attached by peptide bond and arranged like that linearly, right? Without any other modification. That is the linear structure of a protein. Then we have the secondary structure. The secondary structure is when these polypeptides, the protein now, when they are formed, right, they fold, right? The proteins fold into an alpha helix or a beta sheet arrangement. And as you can see there, the alpha helix structures, like if the protein, after they are formed, after the secondary structure, they now take alpha helix shape, like a type of spiral shape, right? Like a spiral staircase. Alpha helix, that is alpha helix, as you can see in the secondary structure there. Let me highlight the secondary structure with another color. This is the secondary structure after primary structure there. That structure there that you are seeing is known as alpha helix or a beta sheet. Beta sheet is when they are just arranged side by side, right? They can either take alpha helix shape or a beta sheet shape. Beta sheet shape is when they are arranged side by side like a side-by-side arrangement, one beside the other, right? That is different from when they form a spiral or alpha helix shape. That is secondary shape. Please, you should be able to know what distinguishes one structure from the other. For instance, we said for the primary structure, there's just when these amino acids are arranged in a linear sequence, one after the other. Like when I start to file you up on a straight line, and I said, as you are coming, you hold your ends. You hold your ends. You hold the end. The end that you are holding together is the peptide bond that is formed, right? So like that, then you form a straight line down to as many number of students that I have, each student representing an amino acid, and your ends that you use to hold one another represent the peptide bond serially, right? That is maybe just a primary structure of the protein. Then the secondary structure is when, and I say, okay, form a straight line. One, you now form columns, right? Maybe 100 on this column, 120 on the other column, right? You still hold your ends there. That is a beta sheet, right? Then the spiral is when you, I don't know, maybe know a spiral staircase. That is alpha helix shape, right? No, it's different from when you just form a straight line beside the form different lines, right? And when there is when you now form spiral, like we ask you to form a kind of a spiral or alpha helix structure, right? That is the secondary structure, right? Then we have the tertiary. The tertiary structure is when the protein now takes a form of a three-dimensional structure folding, 3D, right? For protein to function effectively, this folding is actually important when that protein, after the protein is formed. For it to function, all proteins undergo conformational changes, right? They fold. If they don't fold, they don't function, right? And that 3D folding is known as a as the tertiary structure level, where the protein forms a 3D. I think the keyword there is 3D. The keyword in primary structure is linear. The keyword in secondary structure is alpha helix or beta sheet arrangement. So you can use this to decode what is what at each level. Like I said earlier, the tertiary structure is when the protein takes a 3D conformational change. Then quaternary structure is association of two or more folded proteins to form a multimeric protein. As you can see, when you now have different proteins, like in the after they have undergone, what's it called, tertiary structure, you now have them different protein, like protein 1, protein 2, that have taken their tertiary structure, they now form aggregates, and that is a quaternary structure. And as you can see here in this the third one here, you can see these are different proteins, one depicted by yellow color, the other one depicted by, say, pink, right? No, you now have different protein that has undergone their 3D conformational changes. They now come together in multi-protein, multimeric protein, right? Okay, that is that about the secondary, about the structure of protein. We said there are basically three forms that that protein takes shape. We have the primary structure, the secondary structure, tertiary structure, and quaternary structure. And we've been able to explain or do justice to these structures and how they differ or how they build up from one level to another. Okay. Then we have the nucleic acids. Don't forget, we're treating biomolecules, and we said there are 4 major classes. We have looked at carbohydrates, we have looked at lipids, we have looked at protein. Now we're looking at the last one, nucleic acids. Nucleic acids are molecules that store information, right? And there are basically 2 types. The elements found in nucleic acids are carbon, hydrogen, nitrogen, oxygen, and phosphorus. There are basically 2 types of nucleic acids. You have your deoxyribonucleic acid and the ribonucleic acid. Like we established earlier, the polymers, nucleic acid is a polymer, and the monomers are nucleotide. So what is a nucleotide? A nucleotide is a nitrogenous base, consists of a nitrogenous base, a pentose sugar, and a phosphate group, right? We are now moving from nucleic acids to nucleotides, just as we move from protein to amino acids. We said that nucleic acid is a polymer, right? And they are made up of monomers known as nucleotides. Now, a nucleotide has three major components, right? And they are the nitrogenous base, a pentose sugar, that is a 5-carbon sugar, pentose. Don't forget, in our lecture on carbohydrates, we said that carbohydrates are sugars whose names ends with OSEOs. So when you are seeing pentose from your organic chemistry, pentane, 5-carbon organic, what's it called? Organic compound. Pentose, pent. I think it's a universal. Pent is universal actually. We say pent, hexa, right? Heptane, or like that. Tetrosis, and okay, tetrosis 4, right? So pentose is a 5-carbon sugar, right? And that is the type of sugar that we have in nucleic acid, not trioses, 3-carbon, not exoses, 6 carbons, not tetrosis, 4 carbons. Nucleic acids have a 5-carbon sugar as one of its components. Then a phosphate group, and that is why you can see that if you go back to the elements that make up nucleic acid, carbon, from your pentose sugar, you know there are carbon, hydrogen, nitrogen, oxygen, and the phosphorus. All of these, the phosphate group is also present in a nucleotide. So we are going to see the structure of a nucleotide. Like we said earlier, there are two major types of DNA, nucleic acids rather. We have the RNA and the DNA. We said the sugar that are found in them is a pentose sugar, and this sugar that distinguishes DNA from RNA, or one of the features that distinguishes DNA from RNA. The ribose sugar, the pentose sugar in RNA is ribose, right? While the pentose sugar in DNA is deoxyribose. What is a ribose? What is deoxyribose? We are going to see soon. So that is why we say RNA is ribonucleic acid, right? Ribonucleic acid, whereas DNA is a deoxyribonucleic acid, right? Nucleic acid, ribo, that is ribose, from the ribonucleic, from the word ribose, sugar, right? Then deoxyribose forms deoxy, what is it called? Deoxyribonucleic acid. Okay. Now, why do we say the pentose sugar in RNA is ribose and the one in DNA is deoxyribose? If you look at the structures there in yellow, you see your pentose sugar, right? Okay, maybe I should illustrate. You see this pentose sugar. This is pentose sugar. You see now this pentose sugar, as they have their number with prime, like you have 1 prime. Don't forget, say it's a 5-carbon sugar, and this is 1, 2, 3, 4, 5. That is that in DNA. Then in RNA, you have the ribose sugar with carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Are we together? Now, why do we say the pentose sugar in RNA is ribose and the pentose sugar in DNA is deoxyribose? If you observe these two structures, ribose in RNA and deoxyribose in DNA, look at the carbon 2, where you have 2 prime. That is where the difference lies, right? You see that for RNA, the 2 prime has OH, whereas the 2' in DNA has H. It means that there is no oxygen in at 2' of DNA. No oxygen is bound to this carbon, right? But for carbon 2 of RNA, there is an oxygen and there is an hydrogen, a hydrogen. But in DNA, the oxygen is not there. That's why I say deoxy. That is, the oxygen is removed. D, remover, oxygen, right? So at the second carbon, right, of the pentose sugar, there is no oxygen in DNA. That is why I said there is a removal of oxygen atom. It's deoxyribose. That is a ribose sugar actually, but it is lacking oxygen at 2. And I said that this sugar is labeled prime. Their carbon is labeled prime so that we can be able to distinguish them from the carbon of the nitrogenous base. So when they come together, don't forget we said a nucleotide has 3 structures, 3 components: phosphate group, a pentose sugar, and nitrogenous bases. All these 3 components, they come together in a way, and we are going to see how they pair up, right? The nitrogenous base also has its own carbon. So for us to know that this carbon that we are referring to is for the sugar, not the base, right? So that is why we put prime on the carbon sugar, the carbon atoms of the pentose sugar, right? And in DNA, you'll be hearing 5', 3', 3', 5'. It's so that we are referring to the carbon of the pentose sugar, not the carbon of the nitrogenous base. Okay. Okay, maybe any question? Maybe let's see. We spent 30 minutes talking. Do we have any questions? So I repeat the components of nucleotides. I think I've done that repeatedly. This came in, okay, just now. Okay. Can we call this development stages of protein? No. If you can't say anything, leave and join again. Okay, we'll leave and join again. You're on your own. What is meant by primary and secondary metabolites in my biomolecules? I don't. Why are RNA and DNA called acids? Repeat the components. How is quaternary structure arrangements? Can you take the structure again? Please, we have gone too far. Okay. So many questions. Somebody said, why is DNA and RNA called acids? Because they are acids. Just like you asking me, why is H2SO4 called an acid? Because they are acidic in nature. So they are acids, they are nucleic acids. That's the type of acid that they belong to, right? Nucleic acids. So their names, just like when I say, why is protein called a protein? Why is sugar called a sugar? It's because that is what they are. You can't call sugar kerosene. It's because that is what they are. The organic compounds, the organic compounds, yes, biomolecules are called organic compounds because they have carbon, right? Because their structures are majorly carbon. Why does this difference? Somebody said, why does something different? Why is what different? Why does this difference in deoxy important? Now, somebody asked a very important question, that why is it important? It is important because they do different things. The DNA and RNA do different things. What DNA can do, RNA cannot do it. And what RNA can do, DNA cannot do it, right? And that structure is very, very important to be able to distinguish what DNA from RNA, right? And whether the structure, the presence or absence of oxygen as carbon-2 actually tells us what a DNA is and what an RNA is. And without this, we may not be able to tell and which is which. Okay. What property do they share with acids? What are the three structures? Please, can we now stop the questions, please? I don't know what you mean by what property they share with acids. They are negatively charged. And they are negatively charged. DNA, nucleic acids are negatively charged. I think that is one of the acidic properties that I know that they have. And okay, this is the structure of RNA or DNA, please. We said that nitrogen or nucleic acids has three major components. They are the phosphates, a pentose sugar, and your nitrogenous base. Right? Nitrogenous bases are either purines or pyrimidines. We are getting there. Okay, now please look at this structure very well. You can see that we have carbon 1, carbon 2, 3, 4, 5 with prime. Please know what is attached to what. Like we established in the previous slide, carbon 2, you have either OH or H, right? OH in RNA, H in DNA at carbon 2. Carbon 1 is where you have your nitrogenous base attached. Now we're looking at the three components of a nucleotide. We said nitrogenous base, which can either be purine or pyrimidine. Then carbon 3 is usually OH in both, whether RNA or DNA. That position 3 is usually OH. But carbon 2 can either be OH or H, depending on whether you're looking at DNA or RNA. Then carbon 5 is where you have your phosphate group attached. It is like that in all organisms. Don't ask me why it is like that. Okay, so this is a structure of a nucleotide, right? You have your three major components that make up a nucleotide in this slide. You have your phosphate group attached to carbon 5. You have your nitrogenous base attached to carbon 1. You have hydroxy group OH attached to carbon 3. What tells whether it is a DNA or RNA is what is attached to carbon 2. If it is H, it is DNA. If it is OH, it is a RNA. And this is the structure of a nucleotide, showing the three components, right? Your phosphate group, your nitrogenous base, and your pentose sugar. Okay. Now we mentioned nitrogenous bases introduce purines and pyrimidines. There are two types of nitrogenous bases. We have the purines and we have the pyrimidines. The purines are further classified into two, right? We have adenine and guanine, and we have, for pyrimidines, cytosine, thymine, and uracil. So in all, there are 5 kinds of nitrogenous bases: adenine, guanine, cytosine, thymine, and uracil. In subgroups or subtypes, you have purines and pyrimidines. Out of the 5, 2 are purines, and these are adenine and guanine, while the remaining 3 are pyrimidines, which are cytosine, thymine, and uracil. I hope we get that picture before we say another thing on this slide. Now, we said that one of the differences in DNA and RNA is carbon-2 hydroxyl or hydrogen or OH group, right? Another example in terms of the nitrogenous bases is that in RNA, you have uracil as one of the pyrimidines, whereas in DNA, you have thymine, a T. So, in essence, out of these 5 nitrogenous bases, a nucleic acid can only have 4. In DNA, you have adenine, guanine, cytosine, and thymine. Whereas in RNA, you have adenine, guanine, cytosine, and uracil. There is no uracil in DNA, and there is no thymine in RNA. Thymine is taking the place of uracil in RNA, in DNA, whereas uracil is taking the place of. To easily remember, you can use this. I agree to purify a cute pyramid. I agree. AG for adenine and guanine in purifying purines. CUT for cytosine, uracil, and thymine for pyrimidine for pyrimidine. Or you can just say patch. If you remember patch, PAG, you can say purines are adenine and guanine. Then you know that the remaining 3 pyrimidines. I'm just trying to find a way that can make you easily remember these words, because somebody can ask you which of these is a pyrimidine. Select the odd one out. I put 3 pyrimidines. I put 1 uracil. I put 1 purine. The odd one there will be the purine, right? So please, whenever you're answering questions, look at your question, read them carefully, and what's it called? To be able to answer correctly. Don't just pick your answers. Learn to read. People do say that ability to read and understand instruction is part of examination. But I also say that ability to read and understand your question is the most important thing in examination, because everybody can read, but do you read to understand? You don't just pick your answer, pim at the first thought. Digest your thoughts and synchronize. Okay, which one is the odd? They begin to say, okay, this is in type of nitrogenous base. We have cytosine, thymine, and uracil, and guanine as option D. Definitely guanine does not belong to that place, right? Okay, so as a recap, we said nucleic acids are 2 types, DNA, RNA. In DNA, there is hydrogen attached to carbon 2 of the pentose sugar. In RNA, there's hydroxyl group OH attached to carbon 2 of the pentose sugar. And we said that the pentose sugar accommodates the 2 other components by taking what's it called, nitrogenous base in its carbon 1, pentose sugar in its carbon 5, right? The nitrogenous base attaches to carbon 1 of the sugar, and the phosphate group attaches to carbon 5 on the pentose sugar. And we said that the pentose sugar has its carbon labeled as prime to distinguish them from the carbon of the nitrogenous base. As you can see here, these nitrogenous bases have carbon. Pentose sugar also has carbon. So how do we know which carbon we are referring to? Just put a mark on one of them. Okay, another thing I would like you to take from this slide is that purines are double ringed, while pyrimidines are single ringed, right? You see, these purines, they have 2 rings. Pyrimidines have one ring each. So that is another example between the nitrogenous bases as well. These nitrogenous bases also differ. Purines, adenine and guanine. Pyrimidine, cytosine, thymine, and uracil. And the differences are there. In DNA, the pyrimidine that is common, that is seen in DNA is thymine. The pyrimidine that is seen in RNA is uracil, in addition to cytosine. Right? So your question can come in different form. Can ask you which of these structure is common to both RNA and DNA. And I put adenine, thymine, cytosine. Actually, I put the options. Or which of the pyrimidines is common to both DNA. Oh, please, nobody will ask you to draw nitrogenous base, so please. But if you want to learn it, but nobody will ask you to draw nitrogenous base in exam. But you don't need to cram the structure. Just know that purines are double ring bases, whereas pyrimidines are single rings. Please, you don't need to cram the structure, how to draw it, right? Your questions, your exams are multiple choice. So I cannot ask you to draw, but somebody can ask you question from, at least from this slide alone, I can have like up to at least 5 multiple choice questions from this slide 25 that we need to think twice before you even answer any of them correctly. Okay. Okay, let's look at the structure of DNA. We have how many minutes more? Okay, about 15 minutes, more than 15 minutes rather. Okay, the structure of DNA. These two gentlemen, Watson and Crick, determined the actual structure of the DNA. And they realized that the DNA is a double helix that is made up of a sugar phosphate backbone on the outside with bases on the inside. What that is saying is that you have the phosphate groups are the backbones. They are referred to as the backbone of the nucleic acids, whereas the bases are the inside, and we're going to see that structure soon, right? So you can liken that structure to your ladder, the one you used to climb, ladder, right? The sides, the two sides can be likened to your nitrogen, to your phosphate group, because they form the backbone, right? Then the rungs where you put your legs are the nitrogenous bases that are inside. Then the backbone are the phosphate groups, right? They form the backbone of the DNA. So that, and they said that the DNA is a double helix. There are two, the ladder, one side, the other side. So they are helix, like they form is part